Welcome everyone to this webinar series, which falls under the title Preparing for Beam Time. And this one is organized by two teams at Links. First, the Environment and Climate team, and also by the Heritage Science team. And Lucia Mancini from SAG, Slovenia National Building and Civil Engineering Institute in Ljubljana, Slovenia. She will present today under the title Synchrotron and Laboratory 3D and 4D X-ray Imaging in Heritage Science Applications. And before I hand over the word to Lucia, I can also mention that after Lucia's talk, which will be somewhere between, let's say, 30 to 45 minutes, Chandana here will stop the recording, and then we will open up for, let's say, an informal Q&A session. So at that point, please feel free to activate your cameras and ask questions, let's say, live. In the past, some people have preferred to ask questions in the chat, but then we noticed that sometimes we have to try and interpret the questions. So it's easier for us if you can ask them live, and don't be scared to ask questions live. It's informal and it's more lively and interactive for everyone. And with that, I would like to present, let's say, the bio of Lucia. So Lucia is a physicist and senior researcher, and she's working at the Department of Materials at SAG in Ljubljana in Slovenia. Since 1995, she has been working in the field of synchrotron and lab-based 3D X-ray imaging using non-destructive approaches, and she also did her PhD at the ESRF synchrotron in Grenoble, France. And since then, she has been working as a beamline scientist at Elettra Synchrotron in Trieste in Italy, where me and Lucia were also colleagues for a set of years. And during her time at Elettra, she acquired extensive experience in the application of X-ray computed tomography techniques to material science, as well as to natural and cultural heritage studies. And Lucia also has a strong background in data processing and analysis, both of 3D and 4D images. And Lucia is also part of the Links team Heritage Science, and she's also part of the core group, and she's heading the work package on visualization. And with that, I would like to hand over the word to Lucia. Please go ahead. Thank you.
Thanks for this kind introduction, Manuel. Good afternoon to everybody. Thanks for joining. As Manuel said, oops, it doesn't work. Okay. I don't know why it doesn't work with the, okay. So I'm presently working in the ZAG in Ljubljana, in Slovenia. That is an international recognized organization in the field of construction. In particular, ZAG is the leading institute in Slovenia for building and civil engineering, and the main activities include fundamental and applied research in the field of building materials, but also heritage science and circular economy. And what is normally done is the development of new methods and testing and measurement, but also certification and attestation of conformity of products, materials, and executed works is performed. And also an important part of our activities, training and research of and technical staff, researchers and technical staff in different technical fields. And of course, the participation in the preparation of projects and at international and national level. I had two characterizing working experience, as Emmanuel already anticipated. So I worked before in Grenoble at the Synchrotron, at the European Synchrotron Radiation Facility for my PhD and postdoc. And after I moved to Elettra in Trieste, Italy, where I worked for about 22 years as a beamline scientist in the field of X-ray imaging. I'm also part of Links Heritage Science. I'm part of the core team for the Heritage Science team since last 2023. And in particular, I'm the leader of the working group 5 about visualization. And visualization is an important part of the seminar that I will present. Why is so important? Visualization can in the field, especially in the field of heritage science, that is the field that we will treat today, is important because it's possible to analyze, for instance, archaeological sites or to visualize archaeological finds. And for that purpose, there are several methods available nowadays with modern LiDAR and laser scanners for field survey. In this case, these are a method to analyze large regions of large objects. And in particular, it's possible for this is an example of a church that has been scanned by a laser scanner. But it's also for objects of medium or smaller size, it is possible to use and to obtain very nice results of structural light. And allowing to obtain very realistic representation and with a very high accuracy of the external features of the objects in 3D. And these are other examples. But if you want to analyze, to access invisible parts that are inside these objects, that are not accessible or that are not accessible with structural light or laser or LiDAR, it's needed to use other types of probes, and these probes are in particular X-rays and neutrons. X-rays and neutrons have a complementary nature with respect to different types of materials due to their difference between neutron and X-ray for the attenuation of some elements. And so for that reason, they are used sometimes separately, but more and more nowadays in synergy in different facilities. So, and also for the application in the heritage science, they are very important. Here we see three picture. The first one is a picture of a statue, and the other one are the X-ray and the neutron image of this object. In particular, the first one is a neutron radiograph, and the second one is an X-ray radiograph. And you can see that completely different features are visible, or complementary features are visible with the two probes. And so this is very interesting for many applications. We will focus today on the X-ray part, but take into account that this is one of the topics that we are working with in the heritage science team at LINX, because it's part of the LINX aim, let's say. So in particular, we will speak about X-ray imaging, so X-ray tomography, that is a 3D imaging techniques that allow with a non-destructive approach to obtain not only the external features of the object, but also the internal ones without needing or sectioning or polish or treat the samples. In most of the cases, especially in the case of heritage science, we fully avoid this kind of approach. So we use completely non-destructive approach. And these techniques allow really to obtain not only, as in the case of radiograph, a projection over 2D plane of your detector of whatever is inside your sample, but a realistic representation of the internal distribution of object inside your sample, allowing to determine precisely the real shape and size of the object at each point of the volume that has been illuminated by X-rays, the X-ray source, the X-ray beam. So this is, in this way, the purpose of these techniques is to obtain some virtual sections in the different direction of the space to analyze the volumetric features of our object. To do this, you can use synchrotron radiation. This is a picture of Electra and this is how the typical shape, you can recognize the typical shape of a synchrotron. This is how the internally is done. So as you know, this is synchrotron radiation is generated by particles, in particular electrons, that are traveling at the speed very close to the speed of light in this circular trajectory, and they emit tangentially to this trajectory some synchrotron radiation that is exploited by laboratories that are named beamline to perform experiments of different natures. In particular, we will focus on X-ray tomography, computed tomography, X-ray imaging, that this is, and we can exploit, in the case of synchrotron radiation, the unique features of this of a synchrotron for this technique that are represented by high energy X-rays, nearly parallel geometry of the X-ray beam, very high flux, and the possibility to acquire images with a very high spatial and contrast, but also temporal resolution. And this allows to do high resolution imaging, but in real time and in situ experiment, and also to exploit with a very high contrast detection, the phase contrast imaging modality that we will see some example later on. But in my work since 2002, I started to do a pioneering, let's say, work in order to combine what you can do with the synchrotron radiation beamline, with combining this with laboratory systems, in particular microfocus-based systems, with respect to micro-CT studies. And thanks to that, we could build design and build some different instruments in Trieste, in particular 2 at Elettra and 1 at the ICTP, that is a UNESCO center located also in Trieste. And these instruments, this is the first instrument that we constructed operational since 2005 for users. And here you can recognize the typical structure of a CT scanner, so the X-ray source, the sample stage, and the detector. And the sample stage is constituted by a vertical and horizontal, or depending, of course, you can have different variants, but you can translate and rotate your sample inside the beam. Another instrument that was operational since 2018 is a walk-in hatch that has been also constructed at Elettra, allowing to study larger samples. So the interest to combine and to use these systems in synergy with the CT beamline is that we can have a complementary side of the beam, also complementary energy availability. And typically, we have higher energies with this kind of facilities, but also we can have a fast access for a feasibility test or to prepare experiments. That is really the purpose of this series of the webinar, and to determine the best conditions or the best parameters for an experiment using synchrotron radiation, where typically you have a very short time, a very let's say sometimes unique possibility to perform your experiment. So the use of these facilities allow to well prepare this experiment. But they are also interested in themselves for many types of measurements and analysis. We will see some examples. How it works, X-ray CT, especially when we work in at the micron or sub-micron scale, that what is called X-ray micro CT, that is the technique that we will speak about today. So you record a series of radiographs of your sample that are named projection. Typically between 1,000 and 2,000 projections are acquired for each scan, for each CT scan. And thanks to the application of a mathematical algorithm called tomographic reconstruction, you can reconstruct the axial slice of your samples that are perpendicular, the slices that are perpendicular to the rotation axis of the object. And after that, you can obtain a virtual model of your sample, allowing to see it in a visualization that is called typically 3D rendering or volume rendering, as in this case. This is a coral, an hydrocoral from a museum. But this is not possible also to visualize the object not only as a wall, but also to, once you have the volumetric reconstruction, you can cut or crop your object in all the different directions of the space and to analyze how the object and the different parts that are inside are distributed and determine their size and distance and so on. When we perform X-ray imaging, typically this is a typical image of a beam line, of a CT beam line. This is the beam line at the left, the beam line. Here you have your object, your sample. X-rays are illuminating your sample in transmission geometry. Here you have a detector, a digital detector, and the rotation stage where the sample can rotate, as I showed before. Images can be acquired in two different modalities. The absorption mode, that is the most conventional one, where the detector is very close to your object, allowing to have contrast on the detector due to the differences in the recorded intensity in case different materials are present in my samples. But it's also possible to exploit phase contrast modality. In that case, we are exploiting the phase variation of the X-ray beam, that is an electromagnetic wave, so having a phase and amplitude. And so at the interface between different materials, we will record a phase modulation that if we give enough space to the beam to propagate between the sample and the detector, so if we increase the distance between sample and detector, allow us to visualize some interferences that will be practically visualized at fringes at the border of the object producing these phase modulations. So this means that we can observe phase modulation or enhance the visibility of object simply changing the distance between sample and detector, and these objects will be enhanced thanks to the visibility of some fringes that are nothing more than Fresnel fringes. This is Fresnel diffraction that surround my object. Let's see a real example. This is a simulation, but let's see a real example. So this is a mimosa flower. This is a synchrotron radiation radiograph in absorption mode at 10 kV. And this is the same sample in phase contrast mode, simply changing the distance between contact distance and 50 centimeter in the right image. You see that the very large difference between these two images and the amount of data that you can get. If we increase the energy of the beam from 10 to 25 kV, we can see that the object is practically invisible in absorption, but still can be well visualized in phase contrast modality. So this explains what I wrote in the previous slide, that phase contrast is detectable even when absorption contrast is undetectable. So even an object that is so weak that is not showing absorption, or if the difference between two objects is very small in terms of the density or refraction index, is still possible. It is still possible to discriminate these objects thanks to phase contrast. And this is exploited in many applications and also in heritage science studies, of course. Phase contrast can be exploited not only with the synchrotron radiation, but also with microfocus-based CT scanners at laboratory scale, of course with less contrast than in the case of synchrotron, but in many cases is sufficient for many applications, or at least to determine the best parameters for synchrotron radiation studies. It depends on what you have to do. And in that case, this is an example of an instrument available at ZAC in our institute since 2011. It's a Zeiss X-ray micro CT scanner that is also allowed to work in absorption and phase contrast modality. This is just an enlarged view of the internal part of the sample, where again you see X-ray source, detector, and sample stage. With this kind of instruments, you can reach a spatial resolution up to 1.5 micrometers, working on object of sizes that is maximum 12 centimeters in diameter and 15 centimeters in height. And it's possible to work in static and time-lapse mode. And we have a detector with several optics, allowing to exploit not only the geometrical magnification between due to the distance between source, sample, and detector, and to the cone beam geometry of the source, but also optical magnification thanks to the use of these lenses. Very recently, from May 2024, at ZAC, we installed a new instrument from a RIG solution company that is called EasyTom XL Ultra. And this is a walk-in large cabinet, is a state-of-the-art instrument, very flexible in terms of type of samples that can be scanned and in terms of modalities that can be used for the scanning, allow to scan samples from sub-millimeters to 47 centimeters in diameter and 65 millimeters in height, but to accommodate very large, even larger samples. The spatial resolution that can be reached, the real spatial resolution is up to 400 nanometers. It's possible again to work in phase contrast mode with a maximum source to detector distance of 1.1 meters, and multiple sources and detectors can be accommodated, and static and dynamic studies can be performed up to 10-second scale. Here we see an internal visualization of the cabinet, one sample on the sample rotation state. This is a device for in situ experiments for mechanical testing. When we use the phase contrast modality that I mentioned before, as I told you, you can visualize an object thanks to these fringes that are surrounding your object. But these fringes are a sort of artifact in your images. For that reason, what is typically done is to apply a phase retrieval approach, allowing to reduce or to remove these fringes in order to reestablish the right differences in gray level between the different components of your sample. And this is routinely applied both in synchronous radiation or in state-of-the-art modern X-ray lab CT instrument. Let's see some examples. So in general, when we want to study natural heritage samples or for natural heritage application, what we want to do is to characterize materials that are typically fossils or bio or geomaterial that can be teeth, bones, plants, or like in these examples, insects in amber. And this is, and typically what we want to determine from this study are species identification, provenance and evolution, restoration and conservation purposes are in many cases the aim of the experiments, physical model, to obtain physical models to create replicas using, for instance, 3D printing or other methods, or deformation monitoring in some cases, or microanatomy and visual histology. We will see some examples of these cases. In the case of heritage science, many different materials can be characterized, like stones, wood, clay, or glasses, metals, paper, and so on. But also not only at the level of the materials itself, but also in case of ancient artifacts that are composed sometimes of different, of a combination of these materials. And in both cases, for natural and cultural heritage samples, typically is not needed to any sample treatment, polishing, or sectioning, or this has to be avoided in most of the cases. And what is very interesting is to combine CT methods with the portfolio of different techniques that are allowing, for instance, chemical analysis or other type of analysis, like fluorescence, diffraction, and different types of techniques that are available in other beamlines or laboratories, not only in the same facilities, but also in other facilities. And for that, it's very important the role of infrastructures like LINX or IRIS, that is the European research infrastructure for heritage science, of CERIC, that is the Central Europe Research Infrastructure, allowing to access multi-laboratories and to combine techniques. When we do this kind of experiments, in the case of heritage science, or in general, even for material science, what the user wants is not only to visualize a nice image of your samples, but also to extract some quantitative information about the materials and about what is inside your object. And so it's necessary to use ad hoc software that are for this purpose available, not that can be commercial or research codes that are available to do this quantitative analysis. At ZAG we have different type of different these softwares. that are Aviso, Dragonfly, and Piper 3D. That is a software code that has been developed many years ago at Electra, and is a free of use. But also Dragonfly has a commercial license, but also an academic license, and not it is completely free for academic use or for research purposes. And we will see now some examples of application also about quantitative analysis. This is an example of a study that has been performed on a fossil sample, a lizard, that was completely embedded in a rock. The size of this lizard was less than 15 centimeters in length. And this study was performed by a multidisciplinary international team, allowing for the first time to obtain results about the origin of squamate that are thanks to this fossil that was found in the Italian Alps, in the Dolomites, and this age is originating from the Middle Triassic period. And this 240 million years ago, more or less. We had the chance to publish this paper on Nature, and it was selected for the cover because this was really when it was published, the oldest reptile never discovered. And even if this reptile was discovered more or less 20 years ago and still analyzed with different techniques, it was never possible to extract virtually the fossil from the rock, and so to visualize the part that was embedded in the rock. Thanks to CT, we could virtually extract the reptile from this rock, and we could also analyze the face that was hid to the direct visibility. And combining the microanatomy of this fossil called Megachirella to that of many other reptiles, combining with the molecular data, the morphological ones, we could move back the origin of this evolution in time of 75 millions of years earlier than was previously thought. And this was very important to study the origin of this species. Another important examples in the field of heritage, of natural heritage, are dental remains. Dental remains are usually representing the most common available evidence for the history of extinct species. And what we are usually doing are using these 3D imaging techniques is the study of microanatomy, giving valuable information on the different tissues that are preserved in the structure of the tooth, crowns, and root. And we will see how we can do this using a multimodal and multiscale approach. And this is very important. We will see how. So this is an example of a laboratory-based micro-CT analysis of an orangutan skull. Here you can see we use the voxel size of 42 micrometers. The voxel is a 3D representation of a pixel using for the acquisition of the sample scan. So here you see two 3D rendering of the skull, and here if you look at this movie, you see a sequence of section of the skull where you can recognize the different teeth that are embedded in the bone, in the mandible, and you can see also different other anatomical parts of the skull. But if you want to focus on teeth, is it possible, for instance, to analyze mandibles at a higher spatial resolution than in the case that we saw before? And this is another example also of laboratory micro-CT study of a Neanderthal mandible from Italy, of an immature Neanderthal mandible, because this was pertaining to a child that when he was 2 years old. Here you see these images were acquired at 20 micrometers. Here you see a picture of the mandible, and here on the right you see a 3D rendering obtained with laboratory X-ray micro-CT. And you see only in this image you see the teeth that are already erupted from the bone. But if you change the transparency of the bone, and so you can access also what is internal to the bone, and you can see all the teeth, also the ones that were still not erupted, and you can see all these teeth. And what you can do is to virtually extract these teeth and to analyze them, each of them, in terms of the different tissues that are present in the teeth and important for their investigation. So that is the crown, that is done with enamel, in particular the enamel that is reported in red, the dentin in yellow, and the pulp chamber in blue. But using a multiscale approach, is possible to go to visualize even finer features in this teeth. And using, for instance, the synchrotron radiation, it's possible to analyze a single tooth and to study with high accuracy the shape of the crown and the thickness of the enamel that is here represented in transparent in this image on the left. And this information are really important, because if we compare our sample, this Archi sample, with other samples from different periods, from different species, so Neanderthal, Upper Paleolithic, and Human extant, and you report with the colors, with the code colors, the thickness of the enamel, you see that for sure our Archi samples is Neanderthal, it's a Neanderthal individual. Because the shape and the thickness, the shape of the crown and thickness of the animal is really a fingerprint of the species. But thanks to phase contrast and synchrotron radiation, you can even obtain more, and you can do what is called virtual histology. This means that you can obtain the analogous of what you are doing normally, cutting, sectioning in a destructive way your sample, your tooth, in order to visualize it under your microscope, and to see these lines that are called cross radiation and rhesus lines, that are daily lines or long period lines, that correspond to the growth of the enamel along weeks or along days. So it is possible to, with a very high precision, determine the age of death of the individual, to visualize if there were some interruption in the growth of the animal that could be related to some pathology or some deformation of the tooth. And you can obtain this in a non-destructive way doing CT, X-ray CT in phase contrast mode with synchrotron radiation. On the right, you have the real histological section, and on the left, a section obtaining in a non-destructive way. And you see that the information are practically very close. But you can see even at a higher spatial resolution, you can even visualize the difference between prenatal enamel and postnatal enamel. So this line, this marked line, is the line where of the birth. So this is the line, the zero line of the enamel growth after the birth. So this is because the birth is a very stressful moment for an individual, and this is represented by this very strong line. We did other examples that we can go very fast at that, but what is important is to understand that thanks to this the use of these modern techniques, it's possible to really to analyze many important samples and to attract also different researchers from museums or from important collection that brings to these facilities samples, unique samples. This was, for instance, an example of the oldest examples or tooth restoration never found in the Neolithic period. And on the right, we see an example of the virtual histology of a tooth of a prehistoric fetus that was found with the mother and it was 27 million years old. And so we had the privilege to study these samples, and this also attracted a lot of visibility from newspaper that is and from TV and so on. This from media in general. This is not important for the researcher themselves that wants to be famous, but it's important for the visibility of the institution and also to attract fundings for further research. Okay, this is, I can skip this, but if you are interested, this is the example of the fetus. Yeah, other interesting applications are the studies of bones, in particular for bones, thanks to phase contrast imaging with synchrotron radiation. It's possible to visualize single osteons and lacunae inside the osteon. Osteons are the basic functional unit of the compact bone. And if you see this movie, you see that these are the canals. These are, if you look at the zoom of this element, these are canals, Versian canals, that where nerves and blood vessels are going through. And you have some, all around, you have this distribution of lacunae, of pores, that you can see very well in these images. And you can reproduce in 3D and Versian canals with these lacunae distributed around. This is a complex, a healthy compact bone, but if you have a bone that has been deteriorated, for instance, because is an ancient sample, you can analyze the degradation of this skeletal remains with respect to health bones, but also to compare different types of samples, like in these studies where we compare juvenile skeletal remains with respect to adult remains from burial settings in Italy. In this case, we could practically study discriminate the original lacunae and the diagenetic lacunae due to deterioration of the bone due to this burial in the deposit. And we could analyze and separate and quantitative analyze this lacunae distribution. You can do also dynamic studies in the field of heritage science. This is an example of cultural heritage application in order to understand how ceramic sample is produced or in a dynamic way. What we did was to use a synchrotron radiation and combine it with the furnace that was developed, custom developed at Elettra, and an induction furnace allowing to reach a temperature up to around 1450 degrees Celsius. And what we did was to start from samples at room temperature and to heat the sample at higher temperature, and after, at the end, to quench back to the room temperature. And during this heat, let's say thermal treatment, we could do real-time in situ CT scanning. This is a global view of the sample inside the crucible. And if you look at the magnification of the region that I highlighted in yellow, you see that it's possible to discriminate inside the ceramic matrix these angular quartz crystals and these rounded clay aggregates. And when we start to increase the temperature, we see that the quartz crystals are before still not melted, but clay crystals start to melt. And increasing even more the temperature, we see that both feldspar and quartz are melted. And we see that when we go back and we quench the temperature, we see the formation of cracks and pores inside the ceramic sample. If we look at the 3D representation of the sample, we see a global shrinking of the object that is due to the particle packing that occurs as internal particle water evaporates. This is typical for ceramic firing. But also you see that if we report, separate, or say, in the gray level corresponding to pores and cracks, you see an increase in the amount of pores and cracks in the at different temperature, increasing the temperature, and especially when we go back to the room temperature. Other examples are of in situ real-time studies are the study of consolidant materials to preserve stones or wood or paintings. So these are typically nanomaterials, nanolimes that are used to consolidate these samples. And it is possible to analyze them with CT, and thanks to we can do the imbibition of these materials inside the sample in real time, and to see how the materials penetrate inside the volume and how is filling the pores, and if it's effective or not, and which are, or if it is detrimental for the microstructure. We can analyze all these kind of features and to do quantitative analysis of the distribution of the different phases. Just to summarize and to conclude, I would like to say that laser and structural light scanners, as we saw, are able to survey only what they see, but in order to have access to invisible part, we need to use X-rays or neutrons. The challenge is to use these probes in general with laboratory or large-scale facilities. When we combine multimodal and multi-resolution measurement, is the data fusion. It is also important to analyze the samples when we at different status. When we do, for instance, multiscale experiments, or when we of the same object, or when we do dynamic studies of the same object. So at different parameters, it's important to do data alignment and scaling, and this can be quite an issue in some cases, even due to sample movement. In the case of fossils, we saw that the separation of the fossil from the matrix sometimes is possible, like for Megachirella, but in other cases it's difficult due to the high absorption and the similar density between the fossil and the rock where it's embedded, and also to the geometry that is typically they are flat object, difficult to reconstruct. And also virtual unrolling is another problem. For instance, in the case of papyri, is an open issue, not a problem, let's say a challenge. It is also exciting, I would say, for ancient document on wood support or other types of, let's say, ancient archaeological find. This is an example of papyrus from Ercolano eruption. So you see that is bar samples, and this is a sample that has been sacrificed by the researchers and open to read it. But if you want to avoid to destroy it and to read, it's necessary to do what is called the virtual unrolling, and this has been done on small areas of the papyrus, but it's still an open issue on larger areas, still under progress. And perspectives are to apply advanced visualization tools like mediated reality. In fact, we are also organizing a workshop in March with links that will be held at ZAC on this topic, applied to with mediated reality applied to heritage science. In that sense, AI and machine learning can be applied to help researchers for acquisition, reconstruction, and data analysis, even to solve the challenges that I said before. And it's very important to extend the time availability and feature detection limits and spatial scales. And to that helps a lot a synergy between laboratory-based instrument and large-scale facilities. This is will help a lot and exploit in the best way beam time, but also to extend beam time availability. And last but not least, the use of 3D printing in the field of heritage and natural science is very important for several purposes: to create models, for reverse engineering, rapid prototyping, for restoration purposes, to create replicas of pieces of art that could be lost or deteriorated in time, or to access cultural heritage to virtually impaired visitors, for instance, in the museum. So it's a very important tool. So thanks to all the collaborators of the studies that I presented, and thanks to you for your attention. If you have questions, I'm here.